Okay. All right. Well, welcome everyone. Thank you all for taking the time to join us for our third Revive seminar. I'm very excited to welcome everyone from various parts of the world to this platform where we essentially share insights and discuss important topics related to advanced HIV disease and clinical trials in general. And while this is hosted by the Revive trial team, we still encourage everyone to share the seminar invites with anyone in your network who may be interested in joining. Everyone is welcome to join these seminars. So just before we get going, just some housekeeping rules. So I just want to kindly ask everyone to keep themselves muted throughout the seminar. And then please also just save all your questions for the end, where you can either raise your hand or you can write your question into the chat as we go along, and then we will address these at the end of the presentation. Okay, so our agenda for today. So first, we'll start with an overview of the Revive trial with a focus on the trial progress and recruitment, which will be presented by one of the Revive co-PIs, Sean Wasserman. And then following this, we will have John Eikelboom, our other co-PI, presenting on the topic of the day, large simple trials. And then lastly, we'll have some time for some questions and answers. So once again, just keep your questions for the end of the presentation. All right, great. So first I'll hand over to Sean. He'll give us the Revive trial overview and updates on the progress.
Thanks, Keisha. Hi, everyone. It's really wonderful to see such a great turnout. Welcome to those people that are not investigators on the Revive trial. And for the purposes of just an introduction and a background to like what this actually is for the people that aren't investigators, I've just got one slide to share with you, just summarizing what the trial is and how it works, and then a couple of slides just updating you on our progress. And this will take very quick because the main event is John. I am an infectious diseases physician based at St. George's University of London and at UCT, which is a partner in the trial together with the Population Health Research Institutes at McMaster University. And this is a randomized controlled trial testing azithromycin prophylaxis versus placebo for people with advanced HIV disease. And we've defined advanced HIV disease for this trial as people with CD4 counts less than 100. Anyone basically with HIV and a CD4 count less than 100 who's an adult and has no acute illness will be randomized across the sites, which I'll show you in a minute, to receive either placebo or azithromycin every day for 4 weeks, and will be followed up for the primary outcome of mortality, all-cause mortality at 24 weeks. So we're really interested in seeing whether azithromycin can reduce this high early mortality that we see in this population compared to placebo. And then there'll be a secondary outcome at 48 weeks follow-up. And just very importantly, this is a pragmatic trial. And so it's delivered within routine care and on top of a background of standard of care. So everything that patients would normally get for the advanced HIV disease will be given, including cotrimoxazole and TB prevention and cryptococcal preemptive therapy. But in addition, they'll get the investigational product, which is azithromycin or placebo. And that is the trial. The target sample size is 8,000 patients, so very ambitious. We want to show a clinically significant effect, and that's why we need such a big trial. And that is the reason why we have partnered with experts like John at PHRI, who are very experienced at delivering these sorts of large, simple trials, which John is going to speak about in a minute. This is our progress so far. If you can just advance the slide, Keisha. So we have partnered with 14 countries across sub-Saharan Africa. In the green, you can see the countries that have already been activated and are actively recruiting. We've got actually 5. Botswana sadly had to drop out recently. And the rest of the countries are going to be activated in the next few weeks, which is very exciting. We really need to expand to be able to enroll in this ambitious trial. Next slide. So over 760 patients have already been randomized into the trial in just over a year across these 6 countries now, or actually across 5. And incredibly, almost 90 patients are being randomized every single month just across these 5 countries, and they're doing an amazing job. There are a number of sites, 19 sites across these activated countries that are contributing to the trial already. And as you can see on the right, the data quality is outstanding with almost no missing data. Next slide. This is our projection for enrollment. We expect to, sorry, my computer's about to die. We expect to complete the 8,000 person accrual by November 2027. We're a little bit behind because of different factors. That's the red line, but we are hoping to get back to the blue line very soon and finish the trial on time. It's ambitious, but we are on track and everyone's doing a really good job. So to the investigators on this call, again, congratulations. Thank you for all your hard work and looking forward to many more. So Keisha, that's all I have to say. Over to John for the main event.
Great, thanks, Sean. Yeah, we're definitely looking forward to having more sites up and running. And looks like August is going to be the month for it. So that is very exciting. So as we mentioned, next up we have our main presentation by Professor John Eikelboom, who is a professor of medicine at McMaster University. He's also a senior investigator at the Population Health Research Institute and is co-PI on the REVIVE trial. So over the past 25 years, John has played an important role in multiple trials on antithrombotic therapies, and more recently involved in clinical trials in infectious diseases. He has been supported throughout his career by uninterrupted grant support from peer-reviewed funding agencies and has published more than 800 papers. And since 2014, has been listed annually in the top 1% of cited researchers worldwide. So John, thank you very much for taking the time out to join us today. We are very much looking forward to your presentation. So I'll hand over to you.
John, you're on mute.
Terrific. Well, thank you, Graham, and thank you, Keisha, and good morning, good afternoon, good evening, everyone. Can you see my slides at the moment, Keisha?
We can, John.
Excellent. Well, thank you very much again. And so I'm based in sunny Hamilton, and I am speaking on a topic that is close to my heart. I want to start out by making some disclosures. Well, one particular disclosure. You may be forgiven for thinking that after I finish speaking on large, simple trials, that I think this is the only tool in medicine to advance science. But clearly that's not the case. This is just one way of answering important questions in clinical medicine. It is an incredibly important one. And the concept of clinical trials, of large, simple trials, is highly relevant to the REVIVE trial, which is an example of such a trial. Before I specifically talk about the large, simple trial, I want to go through some fundamentals to provide a balanced sort of background. And when we think about clinical research, we start with this simple idea. We must ask an important question and answer it reliably. And so my agenda, initially, I'm going to talk about what makes an important question, and then how to answer it reliably with a particular focus on the large, simple trial. So thinking about the question, if a question can be reliably answered, it might be important because it provides new information that will change practice. And this is the most important type of question that we can ask in clinical research. And the REVIVE trial fits very clearly under this category, because the REVIVE trial, irrespective of what it shows, will alter practice by demonstrating that azithromycin either is effective and should be used widely, or that it is ineffective and should not be used. The next category of question is one which, if answered, will improve understanding of pathophysiology. Also very important, will ultimately lead to a change in clinical practice, but by itself does not do so. A third category in list of decreasing impact is a question which generates which produces hypothesis generating information. Important, but one further step down. The fourth is a confirmatory study, and perhaps the least important is a study that provides descriptive information only. Now, in reality, the literature is replete. In fact, it's dominated by studies that report descriptive or confirmatory information, the least important types of questions to answer. Now, a little bit more about low impact questions. Low impact questions, or relatively unimportant questions, as I've mentioned, do not change practice, do not provide insights into pathophysiology, do not provide important hypotheses suitable for future testing. One place where they are important is for young researchers setting out in their career, because they need to make a start and they need to get some publications and learn the scientific method. And so there is still, of course, an important place for this, but we aim to answer questions that are high impact, that are the best type of questions. A moderate impact question, it might not change clinical practice, but it can provide important pathophysiologic insights or an important hypothesis that can be tested, and sometimes important confirmatory information. Now, I'm not an HIV doctor, and Graham and Sean, who are here with us, know this way better than I know this, but I took a quick look at the evolution of information in the HIV story. I remember before even going to medical school, the first description of the AIDS epidemic. This was an important study, this was an important report, but clearly it didn't tell us anything about what caused it, and it had no direct impact on clinical medicine, although certainly in terms of public health and epidemiology, it was important. Several years later, HIV was demonstrated to be the cause of AIDS. Again, a descriptive study, a relevant descriptive study, but had no impact immediately on clinical practice. They're sort of the less important, the less good questions, or the good questions, but they're not the very good questions. And here we have a high impact question, a high impact, a very good question, provides insights into mechanisms of disease and or changes clinical practice. And when we go back to the HIV story in the 1980s and 1990s, we had incredibly important studies that described the HIV life cycle and detailed pathophysiology, and then practice-changing studies, randomized trials of AZT. It seems like an eternity ago when I was in medical school. And then in the mid-90s, the finding of highly active antiretroviral therapy saving lives in randomized trials. So that's an overview of asking important questions and subdividing the questions according to whether they're descriptive, confirmatory, hypothesis generating, providing new insights into pathophysiology, or changing clinical practice. The second part of my presentation, and the main part, is how to reliably answer questions in clinical medicine. And a reliable answer is essential if you have a question that's really important, like what causes cancer. That's a great question. You'll never answer it reliably. There are other questions which are not that important, but you can answer them reliably. But clearly, when you have a question that is important, you need to connect it to the most appropriate study design. So what sort of designs do we have? This is really giving some acknowledgement to the fact that clinical research is clearly not the only important type of design to answer a question. There is basic science, there is classical epidemiology, and in most cases of clinical research, basic science and epidemiology will inform the answer to the clinical problem. But in clinical research, it is the clinical problem that drives the question. And the best example of this is clinicians at the bedside faced with questions that they can't solve because they don't have the information. For example, I have a patient with advanced HIV. Their CD4 count is less than 100. I know that this patient can do very well on antiretroviral therapy, but there is a period of risk during the first 3 months during which they could succumb to various opportunistic infections. Should I give them prophylactic azithromycin to protect them against the risk of dying? That's the clinical problem. It's been driven in part by what we know about basic science, about how azithromycin works, about the epidemiology of HIV. Clinical research can include observational studies, such as case series, case control, or cohort studies. But the ultimate design to answer a question of interventions, such as azithromycin, is the randomized trial. So if we go back to our story of HIV medicine, the first couple of advances were based on epidemiology and basic science. Very important, didn't change practice, but these findings certainly further fueled basic science research and clinical research. And it was the clinical research, and in particular, randomization, that resulted in a change in clinical practice and improved survival and quality of life for affected individuals. Well, what's this obsession with randomization? What is the power of randomization? Now, again, you could be forgiven for thinking when you go through the literature, there are so many alternatives to randomization currently being promoted that, well, perhaps doing a real-world study with 500,000 people, much simpler, use large databases, maybe that can replace randomization, producing answers much more simply, much more rapidly, and without all of the challenges that we currently face in randomized trials. I'm going to come back to that issue in a moment. But let's think first about the power of randomization. Now, this is a quote from JAMA 2024 on a study which laments the lack of integration of randomized controlled trials into clinical practice. And the case they make, and the case that I'd like to emphasize, is that the randomized controlled trial remains the most rigorous instrument to determine what works in healthcare. Now, if you believe that, as I do, one might expect that in 2024, we're asking and answering all sorts of questions about what works in healthcare. And indeed, there are many randomized trials being done, but this is a review from 2020 in the Journal of Clinical Epidemiology, where the authors examined Cochrane systematic reviews focused on the efficacy and safety of interventions, of various interventions in clinical medicine. And their conclusion was that only about 10% of the interventions reviewed in these Cochrane reviews had high quality evidence supporting their use. Only 10%. I suspect that in clinical practice, the proportion of interventions for which we have high quality evidence is even lower, because Cochrane reviews only focus on selected interventions. There are more interventions not evaluated by Cochrane systematic reviews, which have even lower or less quality evidence than the ones reviewed. There are simply far too many low quality studies in the literature, and there is still an enormous evidence gap. So what is the unique strength of randomization? Well, randomization is without question the gold standard for identifying causal relationships. And most of what we do in medicine, whether it's a study on etiology, whether it's a study on prognosis, whether it's a study on efficacy and safety, it's all about examining associations and ideally examining the association which is causal. For example, etiology: does smoking cause lung cancer? Does, well, there are so many things there, but does coffee cause pancreatic cancer? There are numerous questions in the literature. To prognosis: does a disease account for an outcome? To intervention: does azithromycin save lives in advanced HIV? Now, why is randomization so powerful? Well, randomization is so powerful because with the simple toss of a coin or a simple computer algorithm, a population of patients that we might consider for treatment with an intervention is divided into groups that are balanced for baseline characteristics that are known, that are also balanced for unknown confounders, so that the only difference between them is exposure to the randomized intervention. Now, of course, we have to acknowledge that post-randomization confounders can arise, and therefore our efforts, well, we can't be complacent in a randomized trial. We must do all we can to reduce the risk of post-randomization confounding, and there are various strategies that we can use to do this. The bottom line is that by randomizing and by minimizing post-randomization confounding, differences in the outcomes between treatment groups can be attributed to the treatments rather than patient characteristics. So that's a simple run through of the importance of various questions, recognizing that there are many different study designs, but our focus here is on the randomized design, which is the best way to evaluate causal association and the power of randomization by dealing with known and unknown confounders. And that brings me finally to the idea of large, simple trials. Now, not all randomized trials are going to be large, simple trials. There are going to be phase 1 studies, which test whether a drug can be tolerated, phase 2, in which we evaluate dose finding. Those tend to be more complex, smaller studies with lots of data collection, with very careful safety reporting, because we know very little about the treatment. But large, simple trials come into their own when treating common conditions, HIV, tuberculosis, malaria, coronary artery disease, common cancers. In these conditions, usually effective treatments have only moderate treatment effects. For example, a 10 to 30% relative risk reduction on hard outcomes that matter for patients, for example, mortality. No one needs to do a randomized trial of the risk of dying with or without a parachute after jumping out of a plane. That is, if the plane is flying, because we know that 100% of people who jump out of a plane without a parachute from a significant height will die. And we know that a very low proportion of people who do so, who are trained to use a parachute, will die. So large, simple trials, we're looking at common conditions where intervention can be expected to only have moderate effects on really important outcomes. To reliably detect a moderate treatment effect, you need a lot of power. You need to randomize a lot of patients with a lot of events, because the signal is small and we need to distinguish signal from noise. That means we need a lot of power. Keeping such a trial simple means that the trial can be large and it can be affordable. And if we had broad entry criteria, the results can be widely applicable. Simplicity reduces costs, and when costs are reduced, we can reallocate resources to activities that ensure the most reliable results. Now, there are many examples of this in clinical research. We did a trial called COMPASS, which tested 2 interventions in people with cardiovascular disease. Although it was a very large trial, it wasn't as simple as we would have liked. It was 27,000 people. But we set out with a sample size of about 19,000. We tried very hard to keep things simple. And then when things were going well and we looked at the event rates, the resources were found to increase the sample size first to about 21,000 and then to 27,000. This was an example where trying to simplify the trial conduct allowed resources to be redirected to activities that ensure the most reliable results. And one of those is randomizing enough patients. Now, what's enough patients? And this is a table that comes from a paper by Salim Yusuf, who used to be the head of the Population Health Research Institute where I work. And it shows some very sobering numbers. Consider for a moment that you have a population with a control event rate of about 10%. And you have, this is in a common condition such as cardiovascular disease or HIV. And the event rate in our REVIVE trial, we project mortality at about 10% in the first 6 months. If we expect only moderate treatment effects, such as a 25% risk reduction, if we were to do a trial where there were between 0 and 50 deaths, which means we'd only get 500 patients, there is a 90% chance that we would never detect, with a p-value of 0.01, we would never detect a 25% risk reduction. Instead, in a REVIVE-like trial, this table tells us that we're going to need at least 500, 600, 700 events. That means at least 5,000, 6,000, 7,000 patients if we are to avoid a 10 to 30% probability of failing to see a significant treatment effect. In fact, we should probably shoot a little bit higher to ensure that we have an adequate sample size and adequate power to detect a 20 or a 25% risk reduction. And in fact, the REVIVE trial was designed to enroll 8,000 patients, as Sean just showed you. We've got a 10% event rate, so we expect about 800 events if all patients receive control, and a somewhat lower event rate, maybe 600 and something, if we expect to detect a risk reduction, or around 700 if we detect a risk reduction of 20%. So they're the sorts of numbers that we need to reliably detect moderate treatment effects on hard outcomes that'll change clinical practice. Now, one of the most important areas in medicine where this has been achieved consistently over the past 30 years is cardiovascular medicine. For those of you who are familiar with cardiovascular medicine and large trials, back in the early 80s, these trials were conceived. The ISIS trial, the TIMI trial, the GISI trial, and many subsequent trials, large, powered for mortality, have changed practice dramatically around the world. And for example, the COMPASS trial had a clear result, and within 2 to 3 years of the result, regulatory agencies in 100 or more countries around the world approved this treatment routinely for people with cardiovascular trials, with cardiovascular disease. But this is only going to be possible if the trial is simple. Indeed, Sir Richard Peto, who's one of the architects of the large, simple trial, made this statement, and perhaps this is the most important statement in my presentation. When we do large trials with interventions that we're familiar with, and we're trying to modify hard outcomes, the trial should be designed so that it is so simple that when we don't know what to do, when there is uncertainty about the correct treatment, it is easier to randomize than not to. So how do we unpack this story? Well, all of us have been at the bedside trying to make recommendations about treatment. And when we know what to do, we say to the patient, we think you should take treatment X because it works, it's going to make you feel better, and it's going to make you live longer. No question. But there are many situations, our patient with HIV, where we've already given them a handful of drugs, and then we wonder, should we or should we not give them azithromycin? So the explanation could be, I think you should get azithromycin, here are the possible benefits. But if you don't take azithromycin, you won't have to have the side effects of this drug. We don't really know what to do here. What would you like to do? And then you end up with a conversation with the patient, which takes you far more time in the ideal world than simply randomizing the patient. So the principles of a large, simple trial, it should be really easy to enroll. Include a very broad population of patients, so you don't have to think too hard. The treatment should be very simple. If we have a known safety profile, we don't have to go into all sorts of fancy safety reporting. Ideally, we want an objectively determined outcome so that we don't need to spend resources on adjudicating events. And we want brief and very focused data collection, which ideally the investigator can complete in real time, or we can extract from the electronic health record. Now, I recognize that each of these items can be, this is a simplification, each of these items can be further considered. It's not always possible to make the entry criteria too broad because, for example, in advanced HIV, if you set your CD4 count eligibility cutoff to below 100, you're going to get higher event rates. If you put it up to 200, easier to enroll, but your event rates go down. You need a bigger study. Not all treatments are simple, and in some cases, the safety profile is less well established, which means that safety reporting needs to be a little more detailed. A blinded trial with objectively determined outcomes rarely requires event adjudication, but sometimes that's not possible. And we do become greedy as investigators because we know that one trial can help to answer more than one question, and therefore often the data collection is more complicated than can be done at the bedside at the first visit. Now, large trials used to be done with perhaps $1 million or $2 million for several thousand patients. But over the past 40 years, there's been a huge increase in costs of doing trials. In fact, some recent trials in cardiovascular medicine are costing in the order of $30,000 per patient to complete the trial. Now, these costs are driven by a lot of factors. One of them is the extensive red tape for study approvals. Now, I only have to think of the Revive trial. Our network has done an incredibly good job. Our national leaders have been very patient in dealing with regulatory and ethics requirements. But if we step back for a moment, we're trying to save lives in HIV with a simple antibiotic. Why should it take a year or more to get a site up and running? Is it really necessary for so much red tape to slow down progress? We have incredibly lengthy consent procedures, which are driven by medico-legal considerations. And I'd put to you that most ethics-approved consent forms are unethical. No one in their right mind will read a 5 or a 10-page consent form. And even if they did, most of our patients wouldn't understand it. But consent forms are becoming increasingly lengthy to serve the lawyer, but not the patient. There is unnecessary routine blood collection. Let's make sure we take a blood sample at the beginning and after 3 months and after 12 months, just in case something might happen. Never happens in routine practice, but because we're doing a trial, there is a requirement for all sorts of safety bloods. On-site monitoring, often a process, very expensive, that can be replaced by remote monitoring. And dare I say, I said here politely, onerous safety reporting, but let me say wasteful and useless safety reporting. A little bit more about those issues in a moment. But as a consequence of these cost blowouts driven by all of these requirements, the recent direct oral anticoagulation registration trials in atrial fibrillation, RELY, ROCKET, ENGAGE, and the Aristotle trial, each trial alone cost in excess of $500 million to complete. No wonder drugs are expensive. So much of what was done in these trials added very little or no value. Is it still possible to do a large, simple trial? Well, it is possible, and I put up a trial that we published in 2016. This was 30,000 patients randomized to different durations of blood storage. We evaluated the effect on mortality. We used electronic databases. We had a waiver of consent. We did the entire trial for around $1 million. So let's look for a moment at some of the myths about randomized trials, and in particular, the myths about what is necessary to do a good trial. The first is the question of on-site monitoring. And if you listen to what your regulators say, we recently had a response from a regulator here in Canada who said you must do on-site monitoring for every site for a large, simple trial that's being done here. What's the evidence that on-site monitoring improves trial conduct or even detects fraud? Well, there is no evidence. It's often required by guidelines and enforced by regulators. But remember, if you do on-site monitoring, you really only detect problems after they have occurred. We now have much better, much more efficient, much less costly approaches to central monitoring using electronic case report forms with rapid feedback to sites. And this is likely much more effective in preventing problems and certainly much more efficient. And we can even review consent forms electronically using the video functions that we currently have access to. So on-site monitoring, very expensive. The CROs love it. They make lots of money from it. Adds very little value. Every trial will see some fraud. Fraud you can't legislate to prevent. On-site monitoring doesn't protect you against the occurrence of fraud. You can sometimes detect it after it's happened. The second big myth, that all adverse events must be collected. I'm going to argue that not even all adverse events need to be collected. And the second part to this myth, that all serious adverse events must be reported to the sponsor and to regulators and ethics committees in an expedited manner. This is absolutely untrue and adds no value. Remember that safety reporting, the goal is not to fill the regulator's inbox with isolated reports. The goal is actually to protect the safety of trial participants. We'd make the argument that adverse events that clearly have no relation to study intervention are not a safety concern, and therefore, unless your drug is stopped because of an adverse event, you shouldn't even bother collecting it. All adverse events potentially related to the study intervention must be collected and reported. That's fair. But in fact, it's not the regulator or the ethics committee that needs that information. It is the DSMB who have knowledge of treatment allocation that are in the best position to protect patient safety. So if we think of a trial like the REVIVE trial, a phase 3 study where the drug adverse event profile to azithromycin is well established, we don't collect adverse events that do not lead to study drug interruption. It's a waste of time. We collect all adverse drug reactions, and we collect all serious adverse events, and we report them all. Serious adverse events that are study outcomes, many people insist that if you're doing a trial of HIV and someone dies, you should report it as a serious adverse event. The trial is testing whether the intervention will prevent those deaths. So it makes very little sense to send individual deaths to a regulator, to an ethics committee. You need to know the randomized treatment allocation. You need 8,000 patients to determine a treatment effect. So why waste our time reporting even deaths to ethics committees or regulators? A pragmatic approach, serious adverse drug reactions that are expected also should not require expedited reporting, but sometimes as a concession to regulators and ethics committees, we agree to do this. It's really only serious adverse drug reactions, so related serious events that are unexpected, that require expedited reporting. And even here, I'm not sure that it adds value. I can't think of a randomized trial ever where a serious adverse drug reaction that is related or possibly related, that was reported in an expedited manner, led to the earlier detection of a problem. Because mostly if these serious adverse drug reactions are reported, the steering committee gets to know about them, the DSMB gets to know about them, and that's where the issue becomes known. The third area, the third myth about randomized trials is that event adjudication is required to produce accurate results. Now, event adjudication, to be clear, in some situations does add value. For example, when it is difficult to diagnose stent thrombosis in a cardiovascular medicine trial, but in most cases adds no value. Indeed, in most cases, it's of unproven benefit in phase 3 blinded trials using hard outcomes. And in fact, it may lead to obliterating a signal rather than enhancing the signal of treatment effect. We all think that event adjudication is to remove events that are not events, and therefore we will detect a true signal. But there's a body of literature that indicates that adjudication actually doesn't help. And here is an example from the COMPASS trial. This is a paper that we published a couple of years ago. The lower curves here, those dotted lines, orange is the control treatment and blue is the investigative intervention, the experimental therapy. So based on the bottom two lines, there was a significant treatment effect, a 24% risk reduction. The top two lines are actually the reported events. The difference between these two sets of lines is adjudication. So we diligently adjudicated as required by the various regulatory agencies and the sponsor. We diligently adjudicated events. That meant we couldn't create new events. We could only eliminate events. We lost 10% of our events. The treatment effect was actually unchanged with adjudication. We lost 1-0 on the p-value. It didn't matter because we had a very powered, well-powered study. But this whole procedure was a waste of time. And for the second intervention, in fact, we also lost 1-0 on the p-value, and we went from a significant benefit to no significant benefit. This has been looked at systematically in various systematic reviews and meta-analyses. This is one example where 47 RCTs involving 275,000 patients the data from these RCTs were pooled, and the ratio of odds ratios for reported divided by adjudicated events was 1. In other words, it made absolutely no difference to adjudicate events. Of course, by not adjudicating, you don't have to submit supporting documentation. Your data are immediately available. You don't have to wait for adjudication. You improve study power and you reduce costs, and any money you save, you put into recruiting more patients. Well, what's the consequence of all this bureaucratic burden and unnecessary trial procedure? You make trials more complex. You increase costs because it's so complex now, you have to have CROs who run the trials. And when CROs run trials, they're here to make money. They will do everything you ask, plus more. They will do lots of safety labs. Everything they do adds to the bill. They'll report all adverse events within the defined time window. The pharmaceutical industry loves them because they do everything by the book. They'll do 100% source data verification. They'll adjudicate all events, but it comes at a massive, massive cost. In contrast, in an academic research organization, we will pay less attention to these things. Our focus is on randomize the patient, keep them on the drug, unbiased event ascertainment and reporting, and follow them all up so that we get reliable results. Now, there are other consequences of this bureaucratic burden and some of the procedures we do in RCTs. Over time, trials tend to get smaller. We are more restrictive in including patients. As a result, the results are less reliable, less generalizable, and there is this shift to observational data sets. And this brings me to the last myth about randomized trials that I want to touch on. And that is that real-world studies provide more efficient and less expensive approaches to evaluating treatment effects. And for this, I'd like to refer you to what I think is one of the most worthwhile articles that you can read if you want to understand randomization versus real-world evidence. And the title tells the story, the magic of randomization versus the myth of real-world evidence. So what is real-world evidence? Well, really, it's just another very big observational study. The strength is you have a lot of patients exposed to a treatment and a lot of patients who are not exposed. It's valuable to pick up treatment patterns, to understand persistence, to detect very large treatment effects, or to detect rare adverse events, because the relative risk for rare adverse events is generally very large. You don't need special safety reporting. When Stevens-Johnson syndrome occurs, there's not too many of those. And if there's 3 in the experimental arm and there's none in the other arm, the signal becomes very rapidly evident. But the problem is that observational studies are subject to bias caused by known or unknown confounders. You deal with that when you randomize. You can adjust and partly account for known biases, but not for unknown biases. And therefore, even very big observational studies cannot reliably detect no treatment effect or a moderate treatment effect. And I'll say that again. If you expect a relative risk in a treatment effect of under 2, an odds ratio of less than 2, a hazard ratio of less than 2, most of our interventions in medicine have a hazard ratio of less than 2, that is in terms of harm, or less than 2 or above 0.5 in terms of benefit. Confounding can never be excluded as a cause for that signal. Now, I have a wonderful example of that, of statin use and cancer-related mortality, but I'm going to skip at that and come to my final couple of slides. And I want to go back to Sir Richard Peto, who said this. Basically, if trials are going to be done, there are lots and lots of questions that could be answered by trials and should be answered by trials and will not be answered by trials unless we reduce the regulatory burden on trials. There are things that we can do. We need better trial guidelines. Most people who write GCP guidelines have never done a trial. It's a bizarre situation that trialists don't write these guidelines, but bureaucrats sitting in rooms who have never seen a patient, let alone run a randomized trial, try and tell us what's good for us. We need strategies to improve recruitment. We need to improve trial quality by focusing on proper randomization, blinding of intervention, maintaining adherence and persistence with drugs or interventions, and ensuring unbiased ascertainment of outcomes. You don't need complete ascertainment of outcomes. You need to get most of them, but it needs to be unbiased, and you need to follow people to the end of the study. I'm going to give you my last slide here. In summary, there are some questions in clinical settings where treatments are expected to have only moderate effects on outcome, on hard outcomes, where we need to do large clinical trials. Those trials need to be simple. They need to be efficiently designed and conducted. We have an enormous challenge before us because there is a tendency to make trials more and more complex, and regulators and ethics committees think it's in their purview and that they're doing a service for medicine by making them more complex. This is a series from JAMA, recently published on clinical trials. It's co-authored by folks from Oxford as well as from the FDA. And this is another paper worth reading. A lot of our practice of medicine depends on the results of clinical trials. but it seems that clinical trials are done in one silo and the practice of medicine is done in another silo. We need to try and integrate these two so that we can achieve the greatest benefits for our patients in the shortest possible time at the lowest cost. Thank you very much for your attention. I'm happy to take questions, and I'm sorry I've gone so close to time.
Thank you so much, John. That was really a brilliant presentation and quite inspiring. And I'm sure it's relevant to everyone on this call. And I'm sure we've all learned something about large, simple trials from your presentation. So if anyone has any questions, please feel free to raise your hand, or you can pop a question into the chat.
So while we're waiting, I see this comment from MPICO saying hello from Pat Comerford there. A couple of the legends of clinical trials in South Africa. There are many there. Hello, MPICO. Hi, Pat.
Great, we have some questions. I see Graham.
Thanks, John, for a great presentation. Thank you.
Oh, there's Pat. Oh, you are there. Hi.
Yeah, hi, John, thanks for a really thought-provoking presentation and challenging. Just one of the issues that you alluded to was that for these large, simple trials, you need a safety record of the drug to really make it appropriate to sort of scale up in the context of a simple trial framework. And I'm just interested if you can speak to that, what degree of safety record you think is drug should be licensed widely, that it should have been evaluated in prior phase 3 trials, or obviously it'll vary according to the context, but just some general principles around that.
Yeah, that's a great point, Graham. And as your question implies, there is a gradient of familiarity and knowledge about the safety of interventions. The less we know about an intervention, the more safety reporting we will require. Of course, that's just one element of making a trial less complex, is reducing safety reporting. I don't think I can offer a clear threshold, but I can make a comment. For example, we're looking at the moment at doing a trial of malaria vaccination. That is, I'm working with some folks who are looking at this in a West African country where the vaccine has not been very well studied. We want to do a large, simple trial, so we're going to do it in 2 phases. The first phase, with a smaller number of people, will have more intensive safety reporting. That'll be overseen by a DSMB, but then there will be a point where a decision is made based on what we've learned to make this much less simple. So I don't think there's any specific threshold. Certainly azithromycin is pretty convincing. Rivaroxaban and aspirin tested in COMPASS were well known, yet we had a regulator at a local site actually reviewing and criticizing us for not reporting constipation as a severe adverse event because someone stayed in hospital longer than usual. That makes it an SAE. SAEs had to have expedited reporting. So that's the other crazy extreme that we've reached.
And John, just one further question on your study, that it's not a phase 2, phase 3, it's just an earlier phase of more intensive monitoring, and those participants will contribute data to the main trial.
That's what we had designed. We haven't got it underway as yet, Graham. That's probably not the traditional model where we separate out the phase 2 and the phase 3. So the other way would be to look separately at the phase 2 to get the safety information ready. In this case, we think that the vaccine is going to be very safe, and we think there is an imperative in terms of time. So we're trying to roll this as a vanguard into the phase 3.
Yeah.
But I don't know that that's appropriate for all types of interventions, especially when safety events may occur later.
Thanks very much, John.
Thanks, John. I think we are running out of time. Let's take one more question and then we're going to have to end the seminar there. So I think Sempagama had a hand raised next.
Have many sites in different continents or countries, and one of the sites or two sites have stock out in terms of one of the regimens. How do you balance with the randomizations? Because you can randomize a participant and pick a regimen which has a regimen is stock out. How do you maneuver that?
If I understand correctly, one of the routine co-interventions has a stock out. If that's correct, I think you raise an important point. When you do a large trial, you have broad entry criteria. Some people would get co-interventions, other people will not. But if you get enough numbers, you'll randomize out any impact of those differences, those background differences. So we'd have to know exactly what situation that applied to. But in general, in a large trial, the variable use of co-interventions is not going to affect the impact of the intervention on outcome in relative terms. You're going to see consistent treatment effects, and it doesn't matter whether those co-interventions vary. I hope I've understood your question correctly.
Thanks.
All right, we are over time. So John, I just want to thank you so much again for joining us and for a really, really great presentation. And thank you everyone for joining our call today. I hope you found the seminar informative. Please do look out for our next seminar, which will be next month. We will send through invitations again, and please feel free to pass on those invitations to anyone that you think might be interested. But yes, thank you all for joining, and thanks again, John.